So I don't know how much people here are aware of the 100,000 Genomes Project. I think most people have heard of it, but not necessarily aware of just how broad a remit this project is. I need to start off with the normal disclaimers. So my opinions are my own, and I have had my travel expenses reimbursed and been paid to speak. Actually, the uses of Illumina products here might not have been approved for FDA clearance. The aim of the 100,000 Genomes Project isn't just to sequence 100,000 genomes. It sounds a bit weird, it's a really large number, but actually it's just a tiny part of the project. We're covering both cancer samples, rare diseases and also infectious diseases. The infectious disease work is being carried out by Public Health England and actually doesn't contribute to the 100,000 genomes. So there will be a database with more than 100,000 genomes in there. But actually the remit is a balance of four main priorities. The first is to bring benefit to NHS patients. So really to embed the programme within the normal clinical pathway within the NHS. Secondly, it's to create an ethical and transparent programme based on consent. So all patients are given a very broad consent, which is both covers both the research and the clinical diagnostic aspects of this project. Also to enable scientific discovery and medical insight. So there is, in fact, a research element to this project as well, and we have a large research community helping us pick out the interesting questions and making sure that we're adding value to the project and really pushing research forward in the UK, but also worldwide. And also to kickstart development of the genomics industry in the UK. And we're doing that both working in pharma and biotech companies, but also working with clinical interpretation partners within the UK and worldwide. So how does the 100,000 Genomes Project work? It was very interesting to hear Edwin's of how his is working. We haven't had the luxury of time to set up small and expand out. So we have 13 of the major hospitals involved that are called the Genetic Medicine Centres, or GMCs as I'll refer them to. And they give complete geographical coverage across the entire of the UK. But around those 13 genetic medicine centres are another 70 hospitals, which are local delivery partners. So we have over 80 contributing hospitals, which mean that there is complete coverage of the UK and there should be very equitable access. I should say England, not the UK. So England, we have got a partnership with Scotland, setting up a partnership with Northern Ireland, and we're hoping to bring Wales in too. So then it will be throughout the UK. But at the moment, this is very England focused. So how does it work? Well, the GMCs actually find the patients that meet our eligibility criteria. They take the patient consent, they take the patient samples and actually do the DNA extraction there. They send the DNA samples into a biorepository once they've got both the samples and the clinical data ready. The clinical data comes directly into the Genomics England database, which is also in England and not in Wales. At one point it looked like it might be. The biorepository are responsible for QCing the samples, plating them up and getting them off to the sequencing centre, which is this lovely building, although it doesn't actually really look like that because there's a lot of building work going on around it. It's in Hinxton in Cambridge. It's staffed and run by Illumina using HiSeq-X machinery, and it is ISO 15189 accredited already, so they achieved that in May. No small task. The data, when it comes back into the database, where it's rematched with all of the clinical data. We also pull longitudinal data, so this is throughout a patient's lifetime, and we'll be bringing it from multiple sources, including GP data. So this will be enriching the entire time throughout the project. The sequencing data is interpreted and actually goes back to the clinicians for validation and feedback to a patient. The GSIP domain. So these are scientific and clinical users of the database who have the interesting questions and can actually help us interpret the more complex cases. And the Gene Consortium, which is biotech and pharma, come into the database, which is pseudo-anonymized, and actually have to do their analysis in there. They can take out the analysis but can't take away the data. And this is very strictly enforced and is part of the patient consent, so that the data never actually leaves the single database. So we had a lot of opportunities mentioned to you in the previous talk, so I'll go through this quite quickly, but actually some of the specific opportunities with 100,000 Genome Project is the fact that this is really being embedded in routine care. So it's collection of the samples through normal NHS clinics. It's part of or becoming part of the normal NHS pathway. There is this single consent for both diagnostics and research use, and we're collecting an interesting set of additional samples, although I was gutted this morning to learn about how interesting platelets were, and my main aim after I get back is to get those in there as well. So we have additional DNA stored for epigenetics, we have tissue and blood stored for transcriptomics, proteomics and metabolomics we're collecting plasma and serum, and for cancer samples we're also collecting plasma for cell-free DNA. We're also working towards a state where we can collect multiple samples from a patient, so be able to take multiple regions of a sample, be able to take multiple tumours, which might be metastases over time, and longitudinal samples. There's a real push to get the high quality clinical data in there and link it to the life course data. And all of our patients are actually consented for recontact so that we can update their reports as we go, and they'll actually be able to access any clinical trials that become available as a result of this. So, very interesting contrast to the previous speaker. We sequence our tumour genomes to 75x. Our constitutional or germline genome is to 30x, and we cover both solid tumour and haematology. So our germline comes from either blood or saliva. We had a lovely explanation of somatic variation, so I won't go through that too much. But actually what we're doing is trying to identify the actionable mutations for reporting back to the clinicians. But actually all of the mutations are left in that database, both from the constitutional variants and the somatic calls in that database for the research and pharma to be able to get to. So where are we within the program? Well, it really kicked off with a pilot phase in 2014, and I will mention the data from those pilots and really focus on the work that we've been doing with fresh frozen and FFPE. They did raise a number of questions that we've been addressing in the experimental pathway, and we see that this will actually go forward probably throughout the lifetime of the project, where we're trying to actually help the pathways become more embedded, enable the GMCs to collect in more different ways, and ensure that the standards of the DNA that we're getting are high. These are our tumour types that are already live. And we've also got a number of approved tumour types coming through over the next few months. That's cancer of unknown primary, childhood and haematological. Taking each one of these live takes a little while because that actually has a custom data model built for each, which helps the GMCs to provide that data that we need for analysis in a reasonable time frame rather than to be too unwieldy. The particular challenges of the cancer program that we see is that it's an incredibly complex patient and sample pathway. So we've got over 80 different collecting centres, all with their own challenges. And no two centres seem to have the same challenges. We're also, if we're ever going to really make a difference to a patient and report back in a timeline that time frame that could change clinical treatment for a patient, we need to be very short turnaround times, and we're not there yet. The molecular pathology and tissue handling is incredibly complex when working with that many sites. We've even found that the pathologist assessment is something that we're having to really focus on training, so that one pathology lab saying that it meets 40% is agreeing with another pathology lab saying it's got at least 40% tumour in there. We focused a lot, and I'll focus the rest of my talk on the processing of the actual samples to make sure they're suitable for next-gen sequencing and whole genome sequencing. But actually, there's also a lot of work going on in the bioinformatics to make sure that we're able to call as low as possible low variant allele frequencies, and also to really get the best benefit out of the CMVs and the structural variants, which is the whole reason why you would do whole genome rather than a targeted panel. So I'll tell you a bit about two of our pilot studies. One was CRUK pilot study, which had 6 collecting centres, and they were collecting just normal FFPE, not doing anything particular, collecting a germline as well. And what we saw was real patterns in sample performance according to the centre. The whole genome sequencing quality was really very affected by both the centre and the tumour type. And what we were really shocked about was actually the sample attrition rate. So they approached 1,601 patients. Of those, over 1,500 actually consented to the program, but by the time we got to the number of samples that we could actually sequence, it was merely 367. So something had gone drastically wrong with samples falling out throughout the QC process. Was the QC process just too rigorous? I would possibly say not. We weren't asking for a lot. We were asking for at least 40% neoplastic cells. Delta CQ is an assay that Illumina have, which looks at the amplificability of FFPE DNA. And then you can actually target the amount of DNA that you need to put into whole genome sequencing dependent on the delta CQ. We were also looking for a 260, 280 of 1.8 to 2.0, so it was actually DNA that we were measuring. So it didn't seem to be too challenging, but yet we were getting this enormous dropout. The second study was a BRC pilot, again 6 more sites, and this time they were collecting triplets, so fresh frozen, FFPE and germline from more than 400 patients. And we collected incredibly detailed metadata about how the samples had been processed and identified, as many of you who work with FFPE know, that there really is no such thing as an FFPE standard protocol. So there were 36 variables between these 6 sites. 19 actually in the tissue processing and 17 throughout the DNA extraction protocol. And this is what we really took into the experimental phase of trying to figure out how do you combine these to get the most likely chance of getting an FFPE sample out that's suitable for sequencing. And good news is that you can do a lot. So one of the first things that we noticed is it wasn't even agreeable between 2 centres of what your whole genome sequencing from FFPE looked like. Up here you've got fresh frozen samples from each site, and particularly look at these regions which are low GC regions, so very AT rich, and a GC rich region. But basically in your fresh frozen sample, you've got a bit of waviness, but it's quite nice even coverage even throughout these regions. One centre suffered from AT dropout, so you can see here that actually the AT regions were undercovered and the GC rich regions were overcovered. But this wasn't even consistent within their own samples. So the first one here looks pretty good. I'd say that looks pretty much like the fresh frozen. While this one here, not so much. The second centre saw exactly the opposite. So they saw GC dropout. So here they've got very low coverage over GC-rich region, but actually AT-rich regions, they've got more reads. So there doesn't even seem to be any consistency between centres of what the problem is. However, the good news was that when we actually really focused on the multivariate analysis to find out the key variables, is that we could improve the quality of the FFPE quite a lot. One of the things that we were looking at was what happens in the molecular pathology lab, how we're fixing it, how long they're fixing it, what fixative they use, which all seems to be really key. But actually, there's also a lot that you can do even on the worst fixed sample. So we found that by dropping the temperature of decross-linking and adding some salt in there, you actually went from this, which I would deny anyone be able to read anything out of this plot here, to something that here looks much more like the fresh frozen material. And we also saw rebalancing of the evenness of coverage and more normalised AT-GC biases. We also took fresh frozen samples from these groups as well, if you remember. So we were hoping that fresh frozen would answer all our prayers. But actually, that's not so easy to do within the clinical normal sample pathway. We found it very, very different for different tumour types and also centres. So some centres already had the collection of fresh frozen material embedded and were very able to do this. Well, some centres, this was a new sample pathway that they were collecting, and it was quite challenging to set it up. There was also a huge discrepancy in the amount of tissue or the number of samples that they could collect with over 40% neoplastic cells. So in the end, we ended up with everything from 68% in endometrial right down to 4% here of samples being able to be used from fresh frozen. We did a number of experiments to see if we could actually help the fresh frozen pathway. And one thing that we had fed back to us was that there isn't always someone available at the end of surgery to be able to do the snap freezing. So our pathologist, Louise Jones, had actually spent quite a lot of time looking at how you could temporarily store fresh frozen tissue to actually hold it before you went and snap froze it. And what she found was 3 conditions. So this is refrigeration in 4 degrees, vac packing like you do meat in a supermarket, either at 4 degrees or at room temperature, can actually really improve things. So here you've got DNA yield, you've got diversity of your libraries. I can't even see that one. One of them is insert size, which I think is this one, and the other one was percent-aligned reads. So you can see that both the DNA quantity and the sequencing itself is very good quality, even if you just put your fresh frozen or your fresh tissue into the fridge. We continue with some experiments on this using cryospray and a number of other techniques to hold the fresh tissue before you snap freeze, because many of the clinics are telling us that they don't even have a fridge, let alone an expensive vacuum packer. The really good thing was all of these storage methods actually maintained not only the DNA and the sequencing quality, but the morphology as well. So they would be suitable for later morphological analysis if required. So just to summarise the pilot findings, we really found that the molecular pathology handling of FFPE is absolutely key. We've come up with some protocols that we have released to all the GMCs and are widely available on our internet site that uses a controlled fixation. It's come up with the idea of a molecular block. So this is a small block of tissue that is handled in a specific way, which is whole genome sequencing friendly. We've actually come up with some recommendations about the DNA extraction conditions for FFPE, which can significantly improve your chances of getting good quality whole genome sequencing. But even then, the FFPE material does still stay suboptimal compared to fresh frozen. Particularly affected is the CMV calling, and this is one of the reasons we want to do whole genome sequencing in the first place. So in our recommendations for the main programme, we said that we preferred fresh frozen material, but where biologically not possible, so meaning they tried, they just didn't get a section which had 40% tumour, they can actually then submit the FFPE material as long as it has been handled in a molecular block fashion as designed in our sample handling guidance. We're monitoring this very, very carefully. So we've started the cancer main program in February 2016, and we're looking very carefully at how that's progressing while we continue with experiments that are aimed to try and help the collection centres really process as many FF samples as possible and as the best quality FFP material as they can. We do these experiments in collaboration with Illumina and also a number of trial sites that we've got. They're mainly the BRC pilot sites that we mentioned earlier. But the focus really is to maintain sample quality while actually increasing the sample numbers that we can get through. One of our big focuses at this moment are biopsies, and it was very interesting what you said about biopsies and that you're already working from these. We're currently trying to work up and start off a pilot using fresh frozen biopsies, which are the diagnostic biopsies. I will tell you a bit more about it. So the consent for diagnostic biopsies is particularly challenging. A patient hasn't yet had a diagnosis of cancer, so how can you possibly consent them for something like the 100,000 Genomes projects? So we've got approved a consent, which is a generic biobank consent to have any excess material put into a biobank. Where possible, and some banks already have this, they can take extra tissue. Where not, it has to be excess tissue. And they have to be able to handle it in the same way as we have described in the molecular friendly fashion. But this has great advantage if we can get the diagnostic biopsies in, because this allows access to people with primary disease who are undergoing neoadjuvant chemotherapy, because at the moment we can only take treatment naive, and this wipes out large numbers of patients. It also gives us access into people with metastatic disease, where we can look at both the primary and metastatic lesions, and also allows us to go for some of these longitudinal samples to actually look at the surveillance. The requirements of DNA are quite challenging from a biopsy, so I'd be really interested to hear in Edwin's viewpoint on how much they routinely get out of the biopsy. We're also worried about reaching this purity measurement and the likely need for macro dissection. But the pilot will really tell us, we've done a lot of literature searching, the pilot really is to tell us how feasible this is going to be. All of the tissue has to be handled in this whole genome sequencing friendly manner, but it also has to be suitable for histology in case the diagnostic block fails. So the cancer programme has taken a phased approach. I've mentioned the pilots, I've mentioned the experimental work that we're doing. I've mentioned the fact that our main programme started in February, so we're down here somewhere in gear one, as we call it, with standard recruitment. So this is broad recruitment of treatment-naive patients. But because Genomics England works fast on everything, we're already setting up, and by middle of June, we should have our first diagnostic biopsies being set up. Our next main targets are to go for multiple tumours, multiple regions, and bring in longitudinal sampling. And these questions have been the specific research questions that the GSIP domains have asked us to address. So they're the interesting research questions. We've really got to cut down our timelines from patient to back to patient report and decrease the amount of DNA that we need to use to be able to get maximum patient benefit out of this. We need to get as many patients eligible and through the door for this as well. And we really need to bring these sampling types in to be able to address the interesting research questions. So what do we see as the legacy of the 100,000 Genome Project? As I mentioned before, it's much more than just returning results to patients. It's about driving a whole transformational change within the NHS. So this really is about developing the structures for collection of tissue within the NHS. It's of sufficient high quality for whole genome sequencing. Getting people used to this broad consent that is being taken for both clinical and research use, and getting the data to be in the same place as the samples. We're also delivering a legacy of infrastructure. So not only is there a sequencing centre, but actually a whole sample pipeline, a biorepository, and a huge data store with all the genotype-phenotype database that is there and sustainable for use in the future in the NHS. We're also concentrating the knowledge of the UK genomics in a single place for both clinical and research. So this will actually really improve the quality of the clinical reports going back, bringing the community together. And it's also one of the first times that we're really trying to push both the NHS, the clinicians, the academics, and the industry partners together and working together in collaboration to look at this data and drive genomic medicine into the NHS. And there's a massive educational programme going on in the UK, both with the general public, with clinicians, with pathologists, with lab scientists, really trying to engage with everybody to up their knowledge about genomic medicine and what it can bring. There is a huge acknowledgements list. I'll have to big up David, who's sitting at the back somewhere. He works with us on the validation, interpretation and feedback working group, and also in the cancer working group. So these guys over here really help us figure out how we report this stuff back, how we validate it, how we interpret it. The cancer working group is helping us decide what the interesting questions are that we should be delivering. The Illumina team, both research and who run the high throughput pipeline. And then I think the biggest thank you of all must go to all the GMCs, because these guys are on the front line. They're consenting patients, bringing these samples in and doing a great job. So thank you for your time.